Welcome to the All Things AI podcast. Today, I'm beyond honored and grateful to introduce Dr. Robert Langer, who is widely recognized as the father of drug delivery, though no title can truly capture the incredible person he is and his accomplishments. He's one of just 9 institute professors at the Massachusetts Institute of Technology and a co-founder of Moderna, which played a crucial role in the global response to the COVID-19 pandemic. In addition, Dr. Langer has received over 1,400 patents, and over the years, his work has been cited over 450,000 times. Simply put, his work has changed the lives of millions around the world. Thank you so much for taking the time to join me today.
My pleasure.
And let's get straight into it. So, a lot of your work has revolved around drug delivery for the past few decades, so I definitely want to cover that in this podcast segment. So, what are the problems in drug delivery that your work has helped to solve? And could you also describe the painstaking role of research in the development of these remedies?
Well, our work in drug delivery goes back actually over 50 years. And the initial innovation was that people never thought you could deliver large molecules like, say, proteins or RNA or DNA. And we developed the first approaches to do that. That was published in Nature, the journal, in 1976. When we did it, people didn't believe could happen.
Yeah.
And so actually, that's evidenced by my first 9 grants getting rejected. But, we did all kinds of studies over time. We got to understand the mechanism. We, brought it into different clinical applications. We created new degradable polymers, new lipids, and we ended up working and starting companies that would use these approaches to treat schizophrenia, cancer, heart disease. It would lead to the mRNA COVID vaccines, and quite a few other treatments. Yeah. Of course, it wasn't just me. It was, thousands of people. A lot of them were my own students, building on the work we did. But and it was over actually a 51-year period.
It's truly incredible, and to see the fact that you've been able to translate everything that's been done in the lab setting to not just publications, but also real world application. I think that's what truly sets your work apart, because it's honestly it's helping people as we speak, and it's just incredible. I think that we're at that point. So I also want to take a second to look more into the process of what the lab work itself would look like. So I guess in the lab, can you walk us through the process of developing biotechnology for drug delivery, maybe through an example?
Yeah, well, maybe that initial example that I just went over is useful. And I think it's really important to realize that we do basic work in the MIT lab, and companies translate that work into real products. So really, what happened, if I start to go back in time, in 1974, I tried to come up with delivery systems for large molecules. And as I mentioned, people didn't think you could. After hundreds of failures and different designs, different materials, we came up with a way to do it. We didn't really fully understand it, but we figured out the mechanisms and even developed mathematical models over time. Then we actually used it for things, again in the lab in animals, with both polymers and lipids for vaccines. Then then the next thing that might be important is when you make nanoparticles, and again, it was kind of basic work. A lot of times those nanoparticles stick together and macrophages eat them. But we came up with special coatings made of polyethylene glycol, which is actually, it's in Miralax if anybody takes that to go to the bathroom. But and that would help prevent those problems. Then we also created some of the lipids. When we use lipids and other people use lipids, might cause toxicity, and they might not get out of a certain cellular compartment called the endosome. So and actually there was also a Canadian group that came up with ways to create what are called ionizable lipids, or ionizable materials. Then even after that, very few lipids had actually ever been created. So Dan Anderson, who was one of my postdocs, now he's a professor, he actually started CRISPR Therapeutics, came up with ways to synthesize thousands of them, greatly creating libraries. And finally, Rohit Karnik, who was one of my postdocs, now professor in mechanical engineering at MIT, came up with ways to manufacture these using microfluidics, all in the laboratory. So then we started Moderna in 2010 with Nubar Fayan and Ken Chen and Derek Rossi, and they took all the things we did and took it far, far further to create lipid nanoparticles, modify messenger RNA, and ultimately that would lead to the COVID vaccine a number of years later.
Yeah, it sounds really, awesome that's how deep this work has gone over the course of the past few years, and there's so many insightful and really impactful discoveries that have came out of it. I also, as I was reading your biography, autobiography from the Kavli Prize, I saw a little bit about, it's not just about you can make the medicines that would or make the drugs that are going to safely be delivered to the cells, but another part of it is also how you're releasing that medicine. So what did the process look like for designing systems that can release drugs in an extremely controlled manner, where you can mitigate factors like the delivery rate of the medicine?
Yeah, well, it all depends on what system you're using, polymers or lipids and so forth. Well, pick polymers for an example. First, it depends on the polymer, and we make degradable polymers, and by changing the compositions, we can make them dissolve at different rates, which will affect obviously the delivery, because then everything escapes. But you can also build in pore structures and other kinds of structures by very simple methods, like changing the particle sizes of the drugs that you put in and changing the amount that you put in, which we call the loading. So, and so you can create different structures that that enable different release rates, and you can even make coatings that'll even further enable you to control it.
For sure. And do you think there's a lot of trial and error that's involved in this process of creating systems for drug delivery?
There is a lot of trial and error in doing it, but you can do what I call intelligent trial and error.
Yeah.
And you can create theories and mathematical models that can really help guide the trial and error. And even now we're looking at artificial intelligence as ways to even further aid in those things.
Yeah, and so you brought up AI, and this is an AI podcast, so I definitely want to cover, I guess, at least a question about that. So how do you foresee drug delivery and discovery being impacted by AI in the course of the next decade or two?
Yeah, it'll accelerate progress. I mean, I think it already has. You know, it's funny, even before people talked about AI or machine learning, what you want to call it.
Yeah.
You know, we started a company, actually, myself and Nick Gallicatos, and Mike Sema was also involved in it, who's a colleague of mine, a professor at MIT, called Transform Pharmaceuticals. This was in 1999. And what our idea was, we would use high throughput screening. We had developed high throughput systems to make new polymorphs, crystal structures of drugs. But also, it turns out then, and I'd say even now, 50% of the small molecule drugs, and most drugs are small molecules, aren't soluble. Mm-hmm. And so what we did is we did thousands and thousands of experiments where we would add different, what I'll call excipients, different molecules, to help them get more soluble. But still, a lot of times we didn't do very well. Yeah. Say you did 2,000 experiments and 3 worked well, but you could take that information and use that for the next generation of experiments, and maybe you'd do better, and you would do better in some, and then you take that information using, again, machine learning, looking at the structures, and do it to the next generation. Ultimately, we would make a lot more drugs soluble.
For sure, and I think I can definitely understand and relate to the part you're mentioning at the end. My project at RSI this summer is in drug discovery with graph neural networks. So I think if you can train the model to look at the structure of these molecules, say, hey, these two might bind together, I think that would be really impactful, because it can reduce the cost of these trials, but also the amount of time it takes, which can be really impactful in finding effective cures.
Absolutely.
Yeah. And so I also want to take some time to shed light on your personal journey through all of this. You've had many different, I don't want to say careers, but I guess you've served as an entrepreneur, you've also been a professor, inventor. So I think there's so much that we can cover in terms of life experiences as well. And earlier you were mentioning, and I read this in the autobiography as well, that you've had many research grants that were rejected. So throughout your career, you've actually faced many people who didn't believe in your work at many different times. So what did you do every single time to ensure that you can continue persevering towards your goals?
Yeah, well, it's depressing sometimes when
Yeah,
people do that. Um, I think I just kept trying. Research at this is a long journey, and you have to keep believing in yourself, believing in what you do. So I would keep trying. I mean, I certainly got discouraged from time to time. I still do, but I believed in what we were doing, and so I kept trying.
yeah, for sure. I think like that's you can apply that same logic to like I guess like a complicated math problem or something, all the way to like the most complicated things in your career. Because I think like the persistence at the end of the day, even if it doesn't yield the exact results you want, you've learned so much through that path. So I think that's definitely some great advice that I myself will apply, and hopefully many people that are watching this. So another part that I was reading about in the autobiography, and something that I believe myself, is that when you like publish something, I think it should actually try and add something to either academia or actually like the real world itself, especially like for discoveries in your space. I think they have the potential to change the lives of many people and really like create more effective cures to many diseases. So what does it take to turn the work and publications that researchers are doing into meaningful application in the real world?
Well, I think it depends on the field. But, in medicine, it takes a very long time. It takes a lot of work. You have to move from what I'll call test tube kinds of experiments, which we call in vitro experiments, to small animals, to large animals, to people. You run into roadblocks, and you keep trying to surmount those roadblocks. So it takes a great deal of work and persistence.
For sure. And what, so I guess like what are some, could you describe maybe like some of the challenges that you yourself have had in terms of like turning your work into something that is actually helping people, for example?
Yeah, well, one is raising money,
Yeah.
but certainly the example at Moderna, when we started it, a lot of people didn't believe that that kind of thing would work, and we had to raise money. We had great CEOs, half a great CEO, Stefan Bancel, and we had Noubar Faye, and they were wonderful that way, and they certainly believed in everything, and did a like I say, remarkable job. We had great scientists who kept walking through walls to get things to work. People like Kerry Benato and Melissa Moore and others, and so we had to get the nanoparticles to work. We had to get messenger RNA synthesized correctly and stable enough, and then there were scale up processes which had never really been developed like that before. So we built a big plant in Norwood, originally to make, you know, personalized cancer therapies, and still will be used for that someday. It's in clinical trials now, but those are so we had to do all those things, and you'd keep running into different problems on all of them, but people kept working hard, and when COVID hit, people would work nonstop, even with lots of protesters outside.
Yeah. I think like it's once again it's a story of persistence, but also it's involved like a lot of the time turning your work in academia into like something that's applicable involves becoming an entrepreneur in the process. That's something that you're definitely familiar with. So what do you think are some of the most successful skills that you've utilized yourself and have seen for becoming a successful entrepreneur?
I think persistence is really important. Maybe dreaming is important. I think those two, dreaming and being persistent enough to keep trying to fulfill your dreams even if you run into obstacles.
Yeah. And so is there like, I guess one thing is, especially like with the amount of startups that are out there today, there's so much that's going on and many companies trying to do like the same exact thing. And so it's like, it's a really saturated field. So is there some sort of like maybe stopping point when it comes to, you're like, hey, like maybe we should move on from this in your own best interest? Or like, is there a way to identify that, or it's, yeah.
Well, I think it's hard to say. First, if people are doing the same things in companies, I think investors will do that for you. What happens is if a field gets too oversaturated, I mean, I think over time it's sort of self-correcting. A lot of money might be wasted, but I think that will be correcting. I mean, the areas all of a sudden get hot, and then people invest, in lots of people, like you say, do the same thing. But then a lot of those companies start doing poorly,
Yeah.
and you know, because they really don't have that much. You know, somebody has to have, I think, a real advantage. Either you have to be early, or you have to, if it's a new area, or you have to, or maybe both, really have something special.
For sure. And so I think it's really, once again, like, it's something that you have to like keep trying, right? Always, no matter what's going on, and then decide based off of that. And lastly, there's a lot of like young listeners that are going to be watching this, I think, generally based on the audience that my podcast has had. So what's one piece of advice that you'd like to leave with the next generation of scientists?
Well, you know, I end up having given a number of commencement speeches, and one time one of my friends heard it, and she actually put it in, I think, 5 words. She said, dream big and never give up," which is kind of what I was saying maybe earlier. That, and what I mean by that is, I think it's really important to dream, not just do things that are incremental, but to do things that really might have a big impact on the world. But then if you try to do that, what'll happen is a lot of people will tell you your idea will never work. And I think you have to really believe in it and believe in yourself, know that you're going to run into obstacles, know that you're going to run into criticism, and keep trying.
Yeah, and I think the dream big and never give up sentiment is something that like I personally can attest to. I've like from the start of high school, that's every single day I've thought that same thing, and it's brought me a pretty far way. So I think that's something that I'm definitely going to carry on forever, and I hope that many people watching this also feel the same way. But I mean, throughout this interview, I've like the shock that I'm actually here right now has hit me multiple times, like in interviewing you in person. But it's like it's truly an awesome experience. I can't describe it with words. So thank you so much for this.
Well, thank you. You did a great job.
Thank you.